So good morning and good afternoon. My name is Thomas Mueller. I'm the Director of Applications for the AFM business at Bruker, and I'd like to welcome you to this installment of the Bruker AFM webinar series. This presentation is titled Investigating Cell Mechanics with PeakForce QNM, and it's presented by Dr. Andrea Slade. Before I introduce Andrea, I'd like to make a few quick logistical comments. First, we encourage your participation during the webinar. If you have a question, please type it into the questions dialog box on your screen. We will accumulate these questions through the presentation, group them together, and then answer them at the end of the prepared remarks. Quite often we have more questions than we can answer in the session, and in those cases we will be sure to follow up with answers via email afterwards. Also, if you'd like to review the presentation afterwards or pass it on to a colleague, the webinar will be posted online within a week in the webinar section of the Bruker webpage. As a follow-up to this presentation, you will also be receiving an email with this link. Finally, when you exit the webinar software, you will be asked to take a brief survey. We'd very much appreciate it if you take the time to complete this, as it helps us pick the topics that are most important to you and to generally make the series better. So let's get started, and let me introduce our presenter. Dr. Andrea Slade is a Senior Applications Scientist in the AFM unit of Bruker's nanosurfaces business. Andrea received her PhD in biochemistry, biomedical engineering in 2004 from the University of Toronto, Canada, where she worked on the integration of optical microscopy and spectroscopy with AFM for the study of biological membranes and membrane protein interactions. Since joining Bruker 8 years ago, Andrea has been focused on life science applications and AFM education and biological research. Andrea has over 15 years experience in biological AFM with over a dozen peer-reviewed publications and has given numerous seminars and presentations worldwide. So let me now turn it over to Andrea to begin this presentation titled Investigating Cell Mechanics with PeakForce QNM. Okay.
Thanks, Thomas, and hello, everyone. To start today's presentation, I'd like to briefly describe the advantages of AFM for cell biology research. First of all, AFM is a single molecule technique. The data obtained is not based on ensemble results, so it really allows observation of subpopulations or individual molecular or cellular structures and behaviors. AFM is also non-destructive to the sample and requires no staining or coating, unlike EM or fluorescence techniques, so native surfaces remain unaltered. One of the key advantages to AFM is that it's operable in a fluid environment. AFM studies can be conducted in water, buffer, cell media, as well as various other types of fluids. And this allows us to conduct live cell studies where we can observe how cells respond in real time to the addition of a drug or changes in the ionic strength or pH of the imaging fluid. Also extremely important to long-term imaging of live cells is environmental control. To maintain cell viability during AFM studies, we can control the temperature of the environment surrounding the cells. We can also perfuse fresh media, as well as maintain a favorable gaseous environment to which the cells are exposed. Altogether, what this really means is that AFM provides a physiologically relevant environment for live cell studies that ultimately allows in situ and real-time observation of cellular structures and processes, both naturally, over time, or in direct response to changes in the local environment. AFM can provide high-resolution, three-dimensional images of live cells. Looking at the AFM image on this slide of human endothelial cells, you can see the high level of structural detail in the cells, like the individual actin fibers, which typically rely on fluorescence labeling to view optically. Fluorescence imaging, however, requires the presence of a certain amount of fluorophore for a structure to be detectable in the image. Excuse me. While this level of labeling may not always be feasible for observing smaller features in a fluorescence image, AFM does not require sample labeling and easily resolves smaller structural detail. And looking to larger size scales, not only is AFM capable of resolving the features of an individual cell, the larger scan sizes afforded by AFM, and for example, the image here is 180 microns, really allows us to observe cell-substrate interactions or cell-cell interactions, which have important implications in tissue structures and healthy versus diseased states. A rapidly expanding area of research in cell biology is mechanobiology. Cells are very sensitive to forces in their surrounding environment. Not only can they sense forces, they're also capable of responding to these forces. The resistance or elastic modulus of a substrate felt by a cell whether that be from a tissue matrix, an adjacent cell, or even a synthetic substrate designed to mimic a tissue surface, directly influences the stiffness of the cell. As such, the same type of cell might exhibit different stiffness or different morphologies when exposed to a softer versus a stiffer substrate. Now, different tissues also have different elasticities. Looking at the top left schematic on the slide, we can see that bone has a higher modulus, with precalcified bone at about 100 kilopascal, muscle at about 10 kilopascal, and brain or neuronal tissue down at 1 kilopascal. So you can imagine that in the development of tissue engineering scaffolds or understanding diseases known to cause significant changes in the mechanical properties of cells, the ability to measure elasticity is very important. There are several ways to measure elastic modulus, including tensile testing or rheological measurements. However, with its ability to measure localized forces over a very small scale, AFM is ideally suited for measuring the elastic modulus of cells and tissues, as well as synthetic cell substrates. To do this, we can conduct AFM force spectroscopy or force curve measurements. Force curves allow quantitative measurement of the attractive and repulsive forces acting on the tip as it approaches and retracts from the surface over a range of piconewtons to nanonewtons. So instead of raster scanning the tip across the surface as we do in imaging mode, with a force curve, we're simply bringing the probe vertically into and out of contact with the surface at a single point. Knowing the spring constant of the probe, the curve is easily converted to a force versus distance curve. Looking at the various parts of the curve can give us quantitative measurements of elasticity or adhesion. We can attach a protein between the probe and the surface and mechanically unfold it. We can also attach a protein or a cell to the probe and then monitor the interactions with a complementary binding surface. And lastly, not only can we measure the forces acting on the AFM probe, we can use the probe to apply a known force to a cell surface and observe how the cell responds to a mechanical stimulus. Now, when we measure cell elasticity or even substrate elasticity, information on elastic modulus or stiffness is obtained from the contact region of the AFM force curve, meaning the portion of the curve where the probe is in contact with the cell surface. Looking at the schematic here on the left, we have the approach portion of a force curve obtained on a cell surface in red, and the approach portion of a curve performed on a substrate surface, such as a glass slide or a polystyrene petri dish, shown in blue. What you can clearly see is that the slope of these two curves is very different. You can imagine that pushing against a harder surface causes more tip deflection than pushing against a soft cell surface with the same amount of force. As such, a steeper or higher slope indicates a stiffer surface or higher elasticity, while a lower slope indicates a softer surface or lower elasticity. Looking to the right side of the slide, we have some force curves taken on 3 different cells with only the contact region of the curve shown. The blue curve was taken on a healthy cell, while the green curve was obtained on a cell that was infected with a bacteria known to disrupt the structure of the actin cytoskeleton. And the red curve taken on a cell treated with the drug cytochalasin, which is known to disrupt actin polymerization. We can see that the slopes of both the green and the red curves are much lower than the blue curve, indicating that disruption of the cytoskeleton changes the cell stiffness, which makes sense, as the actin cytoskeleton is directly responsible for maintaining cell rigidity. As I mentioned previously, forces also play a role in disease states. For example, research has shown that in breast cancer, mammary gland tissue becomes increasingly stiffer during tumor progression. Here on the right, we have some force curve measurement data obtained on a healthy mammary gland. In the optical images, we can clearly see the AFM probe positioned over the lymph node or the surrounding mammary tissue. Force curves taken in these areas, shown underneath the optical images, reveal that the lymph node is much stiffer than the mammary tissue, as indicated by the higher slope of the curve. In force volume mode, individual force curve measurements are performed in a 2-dimensional array over a defined scan area, and the resulting force volume images are essentially now maps of the modulus, adhesion, or even binding interactions with the sample surface. Here we have an example of force volume being used to map the changes in the modulus of a living fibroblast cell as a result of being exposed to the drug cytochalasin, which, as I mentioned before, disrupts actin fiber assembly. In image A, before the addition of cytochalasin, we can see areas of higher modulus, or later colored pixels, that correspond to actin fibers within the cell. At 80 minutes after the addition of cytochalasin, there's a decrease in the cell modulus, with the color scale appearing lower or darker than before. Then at 160 minutes after exposure, the modulus has decreased even further, and we clearly see very little variation in modulus across the cell surface, reflecting the disruption of the actin network. To date, force curves and force volume are the most commonly used AFM modes for measuring and mapping nanometer scale mechanical forces. Unfortunately, though it does provide valuable and reliable quantitative measurements, force volume imaging suffers from slow acquisition speeds as well as low spatial resolution. As a result, this has hindered its widespread acceptance as a standard technique for studying the nanomechanical properties of living cells. In 2010, Bruker developed PeakForce tapping mode. In PeakForce tapping, the AFM probe is modulated at low frequency, at about 1 or 2 kilohertz, using very small amplitudes, typically 100 nanometers or less. As the probe is brought in contact with the sample surface, the feedback signal is the maximum force, or peak force, between the tip and the sample. So if you think about the motion of the probe in terms of Z position, we're essentially performing a force curve at every position on the sample surface. Now, while this might sound very similar to force volume imaging, there are key differences in the operations of these two modes. In PeakForce tapping, we're using sinusoidal ramping and not linear ramping, as is used for force volume or single force curve measurements. With each interaction of the AFM tip with the sample surface, the feedback loop is continuously detecting the peak force. As such, the imaging force control benefits from the results of previous tip-sample interactions. Together with sinusoidal ramping, where the tip velocity approaches zero as the tip nears the surface, this enables PeakForce tapping to directly control the tip-sample interaction force, thereby facilitating the use of low set points, such that imaging forces can easily be kept below 100 piconewtons. This helps protect both the AFM probe and the sample from any potential damage. Additionally, force volume and single force ramps are typically performed at frequencies of about 1 to 10 hertz, requiring significant amounts of time to acquire data. Because PeakForce tapping operates at higher frequencies, we are essentially performing thousands of curves per second. This allows PeakForce tapping images to be acquired at traditional imaging scan rates and at high pixel resolution. PeakForce tapping technology has also enabled PeakForce QNM, or quantitative nanomechanical property mapping. Because we're essentially collecting force curves at every pixel of a PeakForce tapping image, like traditional AFM force curves, we can use these PeakForce generated curves to obtain quantitative nanomechanical property measurements across a sample surface. As a result, in addition to the topographical data, PeakForce QNM simultaneously provides separate and individual quantitative 2D maps of modulus, adhesion, dissipation, and deformation that directly correlate to the topography data channel. One of the important features of PeakForce QNM is that it can be used to characterize a wide range of materials. For example, PeakForce QNM has been used to examine hard materials like polystyrene, pretty much like what you'd find in a standard Petri dish, with modulus in the range of about 2 gigapascal. And at the other end of the spectrum, imaging live mammalian cells having moduli of about tens of kilopascals or even lower. A recently added capability of PeakForce QNM is the use of the Sneden model for modulus calculation, specifically added for imaging biological samples. In addition, we also have the ability to examine and analyze individual PeakForce QNM force curves offline with PeakForce capture, like you can also do with force volume or force spectroscopy curves. And this allows for direct comparison of data contained with the different modes. Here we conducted PeakForce QNM studies of live E. coli cells. The image on the left is a three-dimensional topography image with the color scale representing the measured Young's modulus, with lighter colors representing higher modulus values. We can see that the cell on the right is in the process of dividing during imaging. And looking at the measured Young's modulus, we see that this dividing cell is much softer than the single cell on the left, having an average modulus of about 2 megapascal versus about 15 megapascal for the single cell. What's nice about PeakForce QNM is that PeakForce capture, we can analyze the individual force curves associated with a specific location or pixel in the modulus image. Shown here is the approach portion of the two force curves taken at the locations on the substrate and on the cell as indicated by the two arrows. You can see that the slope of the curve taken on the harder substrate in red is steeper than the slope of the curve on top of the cell in green. Just to note, you can see a change in the slope of the curve performed on the substrate near where the tip first contacts the surface. This happens because the glass substrate is actually covered with a thin layer of gelatin to help the cells adhere to the surface. So what we're seeing at this early point in the curve really is the modulus of the gelatin. However, the thin layer of gelatin becomes compressed as the tip pushes against it, and the tip starts feeling the underlying glass substrate. At this point, the slope of the curve rapidly becomes much steeper as it's now representative of the higher modulus of the glass. As we don't see any change in the slope of the curve taken on the surface of the bacteria cell, we know that this modulus measurement is not influenced by the underlying glass substrate. One other thing to note is the presence of the flagella in the bottom right of the image. That was also detected in the modulus data channel. In this next example, we successfully used PeakForce QNM to look at the modulus of mouse B16 melanoma cells. Imaging was conducted on live cells cultured in Petri dishes and kept in buffer solution during imaging. On the left of the slide, we see a 3D topography image of the lamellipodium of one of these cells. The network of actin filaments is clearly visible in the high-resolution image. Now, this image was collected in about 8.5 minutes. A similar force volume image collected at a ramp rate of 2 Hz with the same spatial resolution would actually take about 9 hours to collect. At the same time that we acquired this topography image, we also collected both the DMT and the Sneden modulus data channels shown on the right. If we look at a single force curve taken in the location indicated by the red and blue circles, we see that the Sneden model fits the curve much better than the DMT model, with the DMT giving us an overestimation of the resulting modulus at 50 versus 37 kilopascal. This is why the Sneden model is preferred for these types of soft samples and is now included in PeakForce QNM. Viscoelasticity is a time-dependent mechanical response to a loading force. The viscoelastic behavior of cells can be studied using the extended range of frequencies or ramp rates provided by combining PeakForce QNM and force volume measurements. Force volume is well accepted as a tool for studying the mechanical properties of cells at ramp rates of less than 10 or 20 Hz. PeakForce QNM can also be conducted at frequencies of 250 and 500 Hz, as well as 1 and 2 kilohertz. The data shown here compares the Sneden modulus maps of the same B16 cell from the previous slide, collected in PeakForce QNM at 250 Hz, with those from force volume collected at 1 and 5 Hz. Comparing the color scale of the cell surface in each image, you can see that all of the maps show that the cell lamellipodium has about the same modulus, independent of ramp rate and technique. Also shown is a plot of the measured modulus values taken from all the image, which shows that for all acquisition frequencies, the peak modulus value is about 20 kilopascal. The lack of variation in modulus values indicates that there's very little viscoelasticity or any other time-dependent deformation mechanism that's active in this range of frequency between 1 and 250 Hz. One thing to note is that force volume images were collected at low resolution, at 16 by 16 pixels. This was done to save time, but the resulting modulus maps do not have enough resolution to clearly identify the actin fibers in the cytoskeleton that are visible in the PeakForce QNM image on the left. Deformability is an important measurement in cell biology. In relation to the onset and progression of disease states, researchers have demonstrated that cancer cells are often softer and more deformable than their non-cancerous counterparts. Glioblastoma is the most aggressive and common form of malignant brain cancer and is one of the most difficult forms of cancer to treat because the tumor cells are resistant to available therapies and because there's few drugs that are capable of crossing the blood-brain barrier to act on the tumor. In this next study, researchers created non-invasive glioblastoma cells by overexpressing tumor suppressive factors, or TSFs, a modification that's also expected to induce changes in the mechanical properties of these cells. Shown here from left to right are the peak force error, elastic modulus, and the deformation images of the non-invasive glioblastoma cells. In the modulus image, darker colors represent softer areas, while lighter colors correspond to stiffer areas. So we can see that the cell nuclei appear as the softest part of the cells, while the underlying glass substrate is very stiff relative to the cells themselves. The deformation image can essentially be thought of as the inverse of the modulus image. The less deformable or darker color an area, the stiffer it is, and vice versa. Both PeakForce QNM and force volume imaging were conducted on a large number of wild-type and transfected cells. As shown by the graph on the bottom of the slide, both techniques clearly demonstrate that the transfection caused the cells to become stiffer and less deformable. One difference between PeakForce QNM and force volume measurements is the statistics provided by each mode. In a specific amount of time, PeakForce QNM can collect a significantly higher number of force curves than force volume. As a result, the standard deviation and the standard error of the mean is much smaller in PeakForce QNM. As such, for these types of studies, PeakForce QNM is more applicable in terms of resolution, quality, and the amount of data delivered. Now, PeakForce QNM can not only be used to map stiffness, it can also provide high-resolution 2D maps of the adhesive interactions between the AFM probe and a surface. So you can imagine that if one were to attach a protein or a small molecule to the end of the AFM probe, and then use this probe to map a surface containing a complementary binding partner that specifically interacts with the molecule on the tip, the resulting PeakForce QNM adhesion map would now represent a map of specific molecular binding interactions, or a molecular recognition map. To demonstrate this, researchers used PeakForce QNM to conduct molecular recognition mapping of the surface of malaria-infected erythrocytes, or IEs, in order to obtain high-resolution images of the distribution of specific molecules expressed on the cell surface. Due to the resolution limitations in AFM imaging of live cells, the presence of these molecules would be quite difficult to check with traditional topography imaging alone. On the top left of the slide, AFM imaging of healthy erythrocytes and malaria-infected erythrocytes with a regular AFM probe shows that not only do infected cells exhibit a very different shape from the typical donut shape of a red blood cell, they also appear to be covered in these knob-like structures that are not evident in the surface of healthy cells. Unlike healthy cells, malaria-infected erythrocytes also exhibit cytoadherence, where they have a tendency to stick to the walls of blood vessels. Not only does this prevent them from being removed from the bloodstream by the spleen, buildup of these cells in the blood vessels can cause other dangerous health issues such as vascular blockages. Molecules associated with the knob-like structures on the surface of the infected cells are believed to be involved in helping them to adhere to the endothelial cells on the blood vessels by interacting with endothelium surface receptors. To examine this further, researchers functionalized AFM probes with the endothelial cell surface receptor, CD36, which is believed to be one of the molecules involved in this cytoadherent interaction, and then used these probes to visualize the distribution of cytoadherent molecules on the surface of infected cells. PeakForce QNM imaging with the CD36 modified probe revealed the areas of high adhesion or high binding affinity, shown as light spots in the adhesion image on the top right, all mapped to knob-like structures in the topography image. The adhesion sites are believed to be specific binding interactions and not just a result of topography variations, as not all raised regions in the topography image correspond to an adhesion or a binding site, as indicated by the green arrow. The 3D overlay image on the bottom shows the co-localization of cytoadherent molecules with the knob structures very nicely. In another example of molecular recognition imaging, researchers used PeakForce QNM to measure the localization and mechanics of individual proteins on the surface of live Saccharomyces cerevisiae cells. Living yeast cells were immobilized in a polycarbonate membrane and imaged in buffer solution. Looking at the series of images on the right of the slide, we see that PeakForce QNM modulus images of the cells, obtained using unmodified AFM probes and shown in the middle left, revealed areas of higher modulus values observed as lighter colors, and that these areas of higher modulus matched to bud scar regions observed in the corresponding topography images on the top left. Comparing these images to those collected with force volume shown on the bottom, we do not see the same level of detail with regards to variations in modulus within and around the bud scar as we do using PeakForce QNM. Now, researchers were interested in mapping the distribution of the yeast membrane sensor MID2, which is a protein on the surface of the cell that serves to detect damage to the cell wall. Yeast cells were genetically modified to express MID2 proteins that protrude from the cell wall and terminate with a His tag, and AFM probes were functionalized with nickel NTA groups such that the nickel was available to bind to the His tag on the cell surface MID2 protein. Molecular recognition imaging conducted on the mother cell wall, the area that's outlined in blue in the topography image, and on the bud scar area outlined in red, found that MID2 sensors were more frequently detected in the area of the bud scar with an average surface density of about 900 proteins per micron versus 300 proteins per micron detected on the native mother cell surface. And this is shown where green pixels in the PeakForce adhesion data channels indicate an adhesion or an unbinding event. The researchers stated that this difference in the observed number of sensor molecules was consistent with the notion that the walls of the bud scars are thinner than the surrounding cell walls, so there's a higher probability for the sensors to reach the cell surface. Control studies were also conducted and revealed a dramatic reduction in unbinding events when molecular recognition imaging was performed using an NTA-modified probe in the absence of nickel, shown in the middle right image. This supports that the observed interactions were specific and not non-specific interactions. Now, because of the high spatial resolution of PeakForce QNM, one of the most remarkable aspects of this study was that researchers were not only able to quantitatively determine the unbinding force of a single nickel mid-2 protein interaction at about 306 piconewtons, but that they were also able to localize the position of these protein molecules with an unprecedented lateral resolution of about 4 nanometers. Bruker's Bioscope Callus AFM is the true functional integration of AFM and optical microscopy, where we can use this multimodal imaging platform for the direct correlation of structure and functional or mechanical properties of biological samples. Moreau, or Microscopy Imaging Registration and Overlay software, allows registration of the optical field of view to the Bioscope Callus AFM scan area. Once registered, optical images can be captured directly into the software environment and then used to navigate the AFM probe to regions of interest on which to perform high-resolution AFM imaging and or mechanical property measurements. For example, here we have two-channel confocal fluorescence image of fibroblast cells. Moreau was used to target the region of the cell in the upper right-left corner of the fluorescence image for AFM imaging. The resulting AFM image is directly overlaid on the fluorescence image. We can then take this region of interest and display the correlated data as both overlaid and individual data sets. In this next study, researchers used combined PeakForce QNM and confocal fluorescence microscopy on the catalyst to study the mechanical properties of living plant meristems. Meristems are regions of cells capable of indefinite division and growth in plants and are usually found in the apical stem and the root of the plant. Meristems were cut from the apex of the stem of an Arabidopsis thaliana plant, and the meristem cell membranes were labeled with a lipophilic fluorescent dye. Anticlinal cell walls, these are the cell walls that are normal to the meristem surface, are thought to be much stiffer than the rest of the cell. In order to investigate this, confocal images such as the one shown here in image A allow easy identification of the anticlinal cell walls within the meristem for selecting specific regions on which to conduct AFM mechanical measurements. AFM topography images of the same region of the meristem outlined in the fluorescence image is shown rendered in 3D in image B, where you can see about 50 cells present within this area. In image C, the same 3D topography image of the meristem cells has been recolored based on the elastic modulus map of the area, where softer areas are darker and again stiffer areas are lighter in color. This image clearly indicates the cell walls are much stiffer than the cell core. While modulus data can be influenced by the topography of the surface, where valleys sometimes appear to be stiffer than peaks because the contact area with the tip is greater, this was not believed to be an issue in this measurement. Green circles show areas where there are divided cells presenting newly synthesized cell walls that are observable in the confocal and the modulus images, but not in the topography image. This type of study illustrates the potential of PeakForce QNM to investigate the mechanical changes of plant cell walls during normal development, as well as in the presence of hormones, drugs, or potentially in a modified genetic background. To examine the differences in the mechanical properties of live and dead bacteria cells, the Bioscope Callus was operated in PeakForce QNM to image the cell surface, while Moreau was used to register epifluorescence images to the AFM images of cells. The bacteria were labeled with a fluorescent live dead assay. This assay allows determination of the viability of bacteria cells by labeling the intracellular DNA of the bacteria or live bacteria with a membrane permeant dye, which fluoresces green, and labels the DNA of dead bacteria with a membrane impermeant dye, which fluoresces red. The idea being that only the dead cells with a compromised membrane will be labeled with the membrane impermeant red fluorophore. Here we see a PeakForce air image of multiple bacteria cells. By AFM topography imaging alone, one cannot easily distinguish between a live and a dead cell. However, with the Moreau registered fluorescence image, shown here as an overlay of the red and green fluorescence data channels, we can easily identify the red fluorescing dead cell at the top of the image from the two live cells that are fluorescing green at the bottom of the image. Using PeakForce QNM, we were also able to map the nanomechanical properties across the surface of the individual cell. Here we have the deformation and the adhesion channels corresponding to the same cells in the PeakForce error and fluorescence images shown in the previous slide, and then shown here in the upper right. The deformation data channel reflects the deformation of the sample surface under the applied force of the tip. Here the deformation image reveals the live cells to have a fairly uniform deformation across their surface. The dead cell, however, appears to be more heterogeneous, with definitive areas of increased deformation, the green-yellow areas, which indicate softer or less elastic surfaces. Interestingly, for some reason, the smaller cell in the upper left of the image was not labeled in the live dead assay. While we were unable to determine if this was a viable cell in the fluorescence images, it would appear from the deformation image that this would be a dead cell, having variable deformation similar to the confirmed dead cell immediately next to it. While the deformation data channel showed distinct differences between live and dead cells, there would appear to be no difference in the adhesion between the tip and the surface of the live or the dead cells, as observed in the adhesion data channel on the right. However, there is higher adhesion between the AFM tip and both types of cells than between the tip and the underlying substrate. Now, not only can the Catalyst generate correlated optical images and high-resolution PeakForce QNM nanomechanical maps, integrating AFM and optical microscopy also opens up a new area of research for optically guided force spectroscopy for conducting localized individual force measurements. This is especially advantageous in situations where nanomechanical data is not necessarily needed or required for each position on a cell surface. In these studies, registered optical images are used to identify structures or specific regions of a cell on which to perform a single or a small number of force curve measurements. The key advantage to this is the elimination of the need to acquire an overview AFM image of the cell in order to locate the same structures or regions. This decreases the time to data and is non-disruptive to the cells. And in cases where researchers are using probes functionalized with a protein or a small molecule, this actually helps to preserve the probe, as the only time the probe-bound molecule comes into contact with the cell surface is during the targeted force measurements. As an example of optically guided force spectroscopy, we'll go back to our example of the live and dead bacteria. We can use the fluorescence image to target specific points in the cells or on the substrate surface on which to obtain quantitative modulus measurements. Force curve measurements were performed at the locations indicated by the crosses in the fluorescent image. A representative curve from the dead cell, shown in blue, the live cell in black, and the substrate surface in pink, are shown in the graph on the right. We see that the slope of the curve on the substrate is higher than that of the live cells, and that both are significantly higher than the slope of the curve taken on the dead cell, indicating that the substrate has a higher elasticity than the live cells, which in turn have a higher elasticity than the dead cells. The difference in elasticity for the live and dead cells is not surprising, as the compromised membrane of the dead cells would result in an inability to maintain normal cell rigidity. And this agrees nicely with our previous QNM modulus images, where the dead bacteria were more deformable or lower elasticity than the live cells. Now, up to this point, I've been showing you how AFM can be used to measure forces. But what about using the AFM probe to apply a known force, say to mechanically stimulate a cell, and then optically detect a cellular response? The ability of AFM to apply highly localized and highly sensitive forces to a sample makes it a very attractive approach to study cell mechanotransduction, the process by which cells convert mechanical signals into biochemical responses. In an example of this shown here, researchers use integrated AFM force spectroscopy and fluorescence imaging to study cell signal transmission in live neuronal cells in order to better understand pain mediation processes. Living dorsal root ganglions were labeled with a fluorescent calcium indicator dye, and the cell soma, dendrites, and axon were identified using transmitted light images to facilitate navigation of the AFM probe to a location of interest on the cell. A tipless cantilever modified with a 10-micron polystyrene bead was used to apply single force stimulations at specific locations on the cells. We can see the position of the tip relative to the axon terminals of a cell in the image on the left. Simultaneously, the intracellular calcium concentration is measured by monitoring the intensity of the fluorescence signal in corresponding fluorescence images. Fluorescence recordings are started before indentation of the probe to quantify basal levels of calcium within the cell. As we see in the fluorescence video, in response to mechanical stimulation by the tip, a corresponding increase in the fluorescent signal is observed due to an increase of free intracellular calcium. Interestingly, the location of the calcium increase was found to occur not only at the tip contact point, but also at points quite distant from where the force was applied, thus proving that the signals are transmitted along the cell. To further study how signals are transmitted from one cell to another, researchers looked at isolated cells as well as cell clusters. As shown in the two fluorescence videos, both primary and secondary stimulations are visible far away from the contact point in both cases, indicating that there's communication between cells even when they're fairly isolated. Additionally, they found that the signal appears to propagate further when the tip applies a higher force, labeled here as hard contact versus soft contact. With regard to studying dynamic processes involved with cell mechanics, traditional AFM imaging has been restricted in this area of research due to the typically longer acquisition times required to obtain a single image, usually on the order of several minutes. With recent advances in high-speed AFM imaging, such as those enabled by Bruker's FastScan Bio-AFM, where images are now obtained in a matter of seconds, we have successfully begun to apply AFM imaging to investigate the mechanics of cell migration. Protrusion formation is one of the essential first steps in this process. The unique combination of high resolution and high-speed AFM imaging has now allowed us to directly observe the formation and advancement of individual lamellipodia and filopodia at the leading edge of live stem cells during migration. Here on the right, we have a FastScan Bio movie of the leading edge of a migrating stem cell. Images were obtained at a rate of 40 seconds per frame at a resolution of 512 by 128 pixels. In the movie, we see two lamellipodia extend in front of the cell, as well as the formation of smaller filopodia. Then, as the cell moves forward, we see a flow of membrane material to the cell front, filling in the space between the lamellipodia. These are very classic stages of cell migration that have been traditionally studied by optical microscopy, like we see in the phase contrast videos of the stem cells on the left of the slide. However, these optical techniques are limited, as they cannot really resolve the small structures, such as the individual filopodia on a single cell. Now, one of the latest capabilities of the FastScan Bio that is very beneficial for these types of cell mechanics or cell migration studies is the new NanoTrack feature, which was released earlier this year. NanoTrack enables tracking of a molecule or a feature of interest in real time. This includes automated scanner offsetting for compensation of any XY movement in order to maintain the position of the feature within a scanned area, as well as simultaneous recording and display of the associated path of movement. In the example shown here, a plasmid DNA was continuously imaged in buffer solution as it was heated from room temperature to 37 degrees. As you can see in the movie on the left, when XY scanner compensation is turned on, the position of the DNA remains stable within the scan area. The movie on the right shows the same strand of DNA imaged during heating without compensation. Here we see how the DNA quickly moves out of the scan area within 2 minutes of imaging due to the drift induced by the change in temperature. With regards to cell migration, NanoTrack could be quite useful in tracking the path of cells, as well as maintaining specific parts of the cell, such as the leading or trailing edge, within a scan area in order to better visualize the nanoscale structural changes that occur during various stages of the migration process. So in summary, as demonstrated in this presentation, AFM offers various modes and techniques for studying the nanomechanical properties of living cells. PeakForce QNM allows nanomechanical property mapping at high spatial and temporal resolutions, while the additional range of loading rates available with force volume can provide important information on time-dependent or viscoelastic properties. Additionally, force spectroscopy can be used to stimulate cells while simultaneously observing corresponding biochemical responses. And together with the Bioscope Callus, Moreau software, and the FastScan Bio high-speed AFM, these advances in AFM to measure, map, and apply controlled forces to cells enables new methods of experimentation in mechanobiology research. So with that, I'd like to thank you for your attention. I hope that this was an informative presentation for everybody, and I think now we'll be taking some questions.
Yes, thank you also from my side, and thanks, Andrea. I see we actually already have quite a number of questions, and more seem to be coming in at a rapid rate now. Let me have a quick look and get started with answering them. I see some of you asked early on whether the slides will be available. So yes, definitely we will. There will be a recording of this available, as well as a downloadable PDF of just the slides. You'll see the link within about a week on our website. You'll also get an email with that link as a reminder. So yes, that will be available. I see also, let me now look at the questions about the content of this webinar. I see an early one on the performing these nano indentations, whether they can be done on live cells, not just on fixed cells. I believe maybe some of this was done on live cells.
Yeah, definitely fixed cells. In terms of AFM measurements, whether it be imaging or nanomechanical properties, are a little easier because they're not moving, they're more rigid, therefore they have a higher modulus. But we have done a lot of work in looking at live cells. So all the data shown on the lamellipodia of the B16 cells, that was live. The glioblastoma cells, they were live. So we really have made a lot of progress in using PeakForce QNM, as well as force volume and force spectroscopy for looking at live cells.
Thank you. Yeah, I see several asked about performing PeakForce QNM, especially on live cells. There are questions on probe choices, MLCT, COD cantilevers, as well as on imaging parameters for obtaining QNM contrast on, and I think it's specifically on live cells.
Right. So I guess there's a lot of definite handy hints and things to look at, and maybe if you can email me, we can get into some specifics. But just in general, with all PeakForce QNM, we want to kind of match the probe stiffness to the modulus range. So you're going to use a softer probe if you're looking at cells down the kilopascal range versus, something up in the high megapascal, gigapascal range. For our data, what we found, the probe we found to work the best was actually the MLCT-D probe, which has a spring constant of about 0.04 Newton per meter. So it's fairly soft, but the other nice thing about it is that it's a short cantilever. Pardon me, because keep in mind in PeakForce QNM, we're oscillating that probe pretty quickly. And in fluid, you can have hydrodynamic drag acting on that probe. So to get a little cleaner of those force curves, we use a shorter probe. So the MLCT-D works really nicely. We've also done some work with the E, especially on the bacteria cells, but the bacteria are a little stiffer. So those would be the two probes I'd suggest.
And yeah, I see there's also maybe a related question on the difference between the DMT and the Sneden model, which may also be relevant to looking at very soft things like
Definitely, yeah. So we did add the Sneden modulus model. You can use it both offline and online. So I kind of showed you in the one example of the B16 cell, we collected both DMT and Sneden. You can collect one, and as long as you have the calibration parameters for your probe, you can actually substitute one for offline. You can apply it to the approach or the retract curve. You can include adhesion. So there's a variety of parameters you can kind of play with that for that. But in general, we do recommend Sneden for these softer samples.
And I see we have a question on comparing PeakForce QNM with force volume, actually, specifically. What advantage does force volume provide that QNM doesn't? Why would you be interested in still doing force volume to obtain additional information?
Well, some people are, as I mentioned, the viscoelastic behavior of cells. Some people kind of like that additional range outside of what PeakForce QNM offers to kind of look at any sort of frequency-dependent or time-dependent behavior, because you have that additional ramp rates. Another reason that we have the ability to directly compare is that, especially when it comes to biological samples, people are very comfortable and pretty much accept the results of the force volume and the force spectroscopy force curve measurements. Those have been around for quite some time. So when you compare your data that you get from force volume directly with what you get for PeakForce QNM, it kind of gives people a better feeling about the results that they're getting with PeakForce QNM.
Let's see, we have one question from somebody. Does the Bioscope Callus provide PeakForce QNM? I believe it's an option on the Bioscope Callus.
Yeah, so like all of our systems that carry PeakForce QNM, so that's the Bioscope Callus, the dimension icon, the multi-mode, the edge. Can you PeakForce QNM on the edge now, the dimension edge?
PeakForce tapping, yes. Adhesion channel, yes.
Adhesion channel, okay. So any of those systems you can get PeakForce QNM, but it's a software key. So it is, if you don't purchase, you can purchase it as an addition to those systems.
And another, I think the user of the Bioscope Callus asks about these capabilities you've shown. Maybe they have an instrument already for a while, whether they can be upgraded to what you've shown. I assume it depends on what. Maybe some of it is a software upgrade.
Yeah. So any of like the addition of PeakForce capture, the additional frequencies like the 250 hertz, the 500 hertz, the longer range. So on Catalyst, because you have a longer range Z, you have the ability to not just do 150 nanometer amplitudes. You can do up to 2 micron amplitudes, so 4 microns peak to peak. So all of those features are part of the nanomechanics package, which are available in probably the latest software version. So if you're not sure if you have the latest, you can contact us and we'll make sure you do.
Yeah, I would definitely suggest to those who wrote that, please do follow up with us so we can make sure you do have the latest software version. You can take advantage of all that. A very different question, functionalizing the probes with proteins, any suggestions or maybe references?
So definitely the group, so the paper that I showed there where they were looking at the yeast cells and mapping those proteins, that was David Alstein's and Yves Dufresne. They have done quite a lot of probe functionalization. So that paper that's referenced on that slide or any of their papers would be very good references. Functionalizing probes is not necessarily one of the easiest things to do. There are groups out there who have seem to make it quite easy and have definitely these protocols that would be useful to follow. But one of the difficulties in doing these functionalization is actually confirming what you have on the end of your probe. So how do you determine that your protein is there? How do you determine that it's in the right orientation? So it's definitely an area that's developing, but as I mentioned, looking to what these groups who have done it for quite some time, what they're doing would be very helpful. Also, we have, if you go to our website, just remembered we do have an app note kind of giving some of the general chemistry protocols that people are using. So you might want to take a look at that on our website. So if you go to the Bruker.com website and go to say the Bioscope Callus, you can look up application notes, and it will be there.
One question about the fast scan bio, how fast it is, what the fast scan bio is.
How fast can it go? That's a great question, and it's one that everybody should be well educated in looking at these high-speed AFMs. How fast this system can go? Okay, so fundamentally, it really depends on your bandwidth of your system. So sorry. I'm doing something on my computer. So the FastScan Bio has higher bandwidth. It requires using these small cantilevers. So you know the cantilevers we're using here are these the Bruker's own FastScan DX probes. So they have spring constant of 0.2 Newton per meter. And their frequency in fluid is about, for tapping mode, is about 100 kilohertz. So, you know, they're pretty high. The other thing to look at when you're looking at resolution or how fast something can go is the number or the pixel resolution when you're acquiring. The lower pixels you're collecting and the smaller scan sizes, of course, the faster you can go. So to kind of give you some general numbers that we found on the FastScan Bio, we have collected those DNA plasmid images, so I showed you with the NanoTrack. We have gotten those images at 3 frames per second. We have imaged those cells. That was 40 seconds per frame because, of course, you're going from 500 nanometer image on the DNA, which we can also go to a lower pixel resolution, to a 10 micron image where you have a cell which has much more topography. And we collected those images at 512 by 128 pixels. So 40 seconds is still actually quite impressive when you look at what it would take you to do on a normal AFM, probably 40 minutes to get, or 30 minutes to get those images. We've also looked at bacteria cells where we're getting 18 seconds or 8 seconds per frame. But in general, if you're looking at molecular resolution, scan sizes less than 1 micron, 1 frame per second is probably the general. It's a long answer, but it's an important one.
Thank you very much. A question on probe choice for looking at mechanical experiments, mechanical probing on fibroblasts in particular. What's the best probe, best medium, role of temperature control? How about spherical probes?
Spherical probes are really nice. So, and a lot of people are using them because you have a very well-defined interaction surface or contact area with your sample. So the models that we use, the Sneden model, the DMT modulus, hold a lot better because you have a very well-defined contact area. If you're looking for high resolution like you are on PeakForce QNM, of course, if you stick a 10 micron bead on the end of your probe, you're not going to necessarily get high resolution. But if that's not what you're necessarily looking for, if you're more really concerned with the nanomechanical property measurements, then those probes are great. In terms of spring constant, for PeakForce tapping, because of that feedback control, we can use slightly stiffer probes than we do for straight force spectroscopy or force volume. Force volume, force curve measurements, you're typically looking at 0.01, 0.06 Newton per meter. And again, with PeakForce tapping, we're getting really nice modulus data when we're using probes that are 0.04 and 0.1, I believe, is the other Newton per meter.
There's another question on the mechanical probing, especially we're looking at modulus, looking at elastio modulus on biosamples. The question whether indenting the cell at the rate of 250 kilohertz wouldn't change its properties. I should maybe note that in PeakForce tapping and some of what we showed, we were talking about 250 hertz, not 250 kilohertz.
Yeah.
So it wasn't as fast a rate as that. But,
yeah.
I mean,
sorry, I might have said kilohertz maybe when I was speaking, but the slide, yeah, it's 250 hertz, as Thomas said. But even at 250 hertz, that's an interesting question because there are, and this is why we kind of have the kind of group of techniques, force spectroscopy, force volume, and PeakForce QNM. Some researchers are very conscientious of they want to collect and only want to touch the cell one time because they do believe that the cell, what they're interested in, the properties of the cell are going to change afterwards. But what we have found when we do our PeakForce QNM data is if we take consecutive images of the modulus and we can compare those images, we don't have very much variation from one to the next. So that would suggest that we're not necessarily changing the properties of that cell. But if it is something that you are concerned with, the ability to use Moreau and do those selected points, you could do 1 point on a cell, and we do have groups doing that where they just are interested in statistics. So they'll take 30 cells or 60 cells and do one curve rather than doing 60 curves on one cell.
Okay, thanks, Andrea. I see we do have actually a lot more questions, and there are more coming in. We are running out of time. So we will follow up on the remaining questions with email, and don't hesitate to contact us if you think of additional questions in the meantime. You'll also get the email with a link to the webinar. So let me thank you all again for attending. I hope you found this webinar very useful. Thank you, and we'll hope to see you soon again for the next one. Thank you. Bye-bye.
Thanks.